ancestry individuals, which represent a very specific moment in time in Charleston's history, being around the Revolutionary War era or the latter quarter of the 18th century, compared with contemporary populations of African and African-descended individuals in Charleston. This is really important also in light of Charleston's history being a largely transitory place. So this will be able to tell us, by comparing the genetic diversity of the ancestry individuals with the individuals that we are involving in our contemporary genetic diversity study, how has population movement in Charleston changed since that later half of the 18th century. Also really important is the community aspect of this project. So in this project, we're hoping to see and hoping to use DNA as a means to engage and start a dialogue with the community about Charleston's role in the slave trade and the legacy of African and African-descended persons in Charleston today. And I feel like this is a really important part of this project. And moving forward, when I talk about the process of that, where you take the ancestry individuals through, I kind of operate on a mindset that these are individuals. They're not samples. They're not data to be processed or sequence data to be obtained. These are ancestors, and the utmost respect goes into them when I'm handling and working in the lab. So I hope you guys know this going forward, because we're going to show some photos of basically the process that's been ongoing for the past couple of months. So back in 2018, when we were here originally, let me stand out of the way, we collected DNA samples from Brockington Associates, which is the archaeological team that excavated the burials. We collected 2 samples per individual in order to increase study success. So here we went individually through each of the boxes. So this is at Brockington Associates, and each of the parts were in, each of the individuals were interned in different bags. And so we would go through each of the bags, examine this with a tooth, it's really hard to see, for preservation, to collect DNA samples. So when we selected DNA samples, we took, basically it was by individual. So each individual you saw with Dr. Abel had variable preservation. So some were really great and some were not so great. We preferentially sampled for what is called petrous bone. Petrous is basically, if you look at your temporal bone, which is right here, it contains your inner ear bone, so your cochlea, right? So the cochlea, or the cochlea, contains the densest bone in the human body. And so the denser the bone, the better the DNA preservation. So this is a new technique that's been developed over the past 2 years, and so we preferentially sampled for this because it yields better DNA, and so increasing the study success. If petrous was not available, we also sampled from teeth, so we took from the tooth roots, which are also really great, great for DNA preservation. Those weren't available. We also took from long bones, metatarsals or metacarpals, which are your finger bones as well. So we took these samples to the University of Tennessee, where I've been living in the last 2 months, to do research today. So University of Tennessee is in Knoxville, so pretty, 5 hours away. They have a brand new DNA laboratory that was just built. This facility is run by Dr. Graciela Cabana, who's been housing and sponsoring this research project, which is really wonderful. And it contains 2 DNA labs. One is a modern DNA lab, so this is what we would have at the University of Pennsylvania, where we can do mitochondrial control region sequencing, very standard DNA lab. And it also contains an ancient DNA clean lab. And so there are maybe a dozen of these in the United States. So, and research like what we're doing with ancestry burials has to be undertaken in an ancient DNA lab. And this is because this lab is free of any sort of modern contamination. So when we do this analysis, I have to suit up, and these suits right here, head to toe, hardly, and no skin showing or exposed, to make sure that I'm only extracting the DNA that's coming from the ancestry individuals and not my own DNA or anybody else's. So we use a lot of bleach to make sure things are clean. We UV the lab every night. It's a full setup, so it's quite a process. So now I'm going to kind of take you through what we take with the ancestry individuals, how we go from bone that Brockington Associates collected to DNA, how do we take that DNA and prepare it to be going to be sequenced. So the photos are going to be a little fuzzy because I had to take my phone in a plastic bag to take photos, so I didn't contaminate the samples with my cell phone. We basically remove excess bone in this, what is called like a bone clone zone. So basically it's a box, so it basically controls any sort of contamination. This is a temporal bone, so that was that petrous portion that I was talking to you guys about. We remove all the rest of the bone that we don't need. So basically we end up with something that looks like this for the petrous bone. And it kind of looks like a little bit of like an octopus almost in a way. So once we've prepared the sample, we basically remove all the surface contamination for it. So we wipe it down with bleach, soak it in water, we UV it. So this basically removes any sort of surface DNA that could be present on the bone. Then we have to take the bone from a solid substance to a powder, and the powder allows us to actually do the DNA extraction. And we do that by using, it's called a freezer melt. So basically it uses liquid nitrogen, which is right here. Very, very, very cold, very fun to do. And you put it in the freezer melt and it freezes the sample, and then which allows a magnet to go back and forth very quickly to grind the sample into a very small fine powder. So this whole process going from bone to powder to do DNA extraction takes me about 4 days per 10 samples. Now we actually get to extract the DNA. So DNA in the powder goes into what is called a cell lysis solution. So this solution, basically, lysis means to explode, right? So it basically breaks open all the cells to remove all the DNA in it. And I love this photo because here's each of the first 10 of the ancestry individuals, and I love all the different shades of bone that you see in there. We take the DNA and we extract it using silica spin column technique. And basically what this means is that we put the solution with DNA and all the proteins and everything that's in the bone into a spin column, and when we spin it really, really fast, it puts the solution through the tube, and the DNA basically gets stuck in the silica membrane. So that way, we're able to remove all the extra stuff, all the stuff we don't need, all the salts, all the proteins, and when we're ready to extract, we basically put another solution that will then release the DNA into the solution. So we're able to basically take all the proteins, all the stuff we don't want, and only isolate the DNA. This takes about 2 days to do. Now that we have the DNA, that's great. That's wonderful. Sometimes we can't get DNA out of it. Now we have to prepare the DNA for sequencing. So once you have the raw DNA, you can't just put it in a sequencing machine to get data. You have to basically put on these little tags at the end of the DNA so that when you put it in the sequencing machine, it allows the sequencer to recognize it. So this step takes about 2 days, and we can do that in our modern DNA laboratory so we don't have to suit up in the full body suit. After this, we submit for a quality check. So remember the ancestry individuals and also common with archaeological individuals, the DNA can be degraded. I mean, it's been sitting in the ground for 100, 150 years. So it's usually sometimes not very fresh, and sometimes it cannot be very good. So we always want to check for the quality and the concentration of the DNA. So this looks a little bit complicated, but I promise it's not. This is called a bioanalyzer DNA assay, and it basically looks at DNA concentration. So here we can ignore this peak. This is size standard, this one as well. So these are just basically for the instrument to recognize. This big peak here that you see is basically a primer. So this is left over from sequencing preparation. What we really care about is this little lump right in the middle. This is actually our DNA. Now the taller the lump, the better it is, because this represents concentration. So we want to see this lovely little peak around 300 that slowly kind of grows out. So this is a really great sample. This is one of our samples, number 12. So where does the project stand right now? So over the past about 4 weeks, I've been able to successfully extract 20 samples. We still have the remaining 16 to go. Of those 20 samples, we have 15 that have passed that quality check, which is a really great sign. I was expecting pretty variable preservation due to the morphological preservation of it. So to have 15 of those samples be decently have good DNA in it is a really good sign moving forward. And there's been no contamination thus far, so it's really great because that means I'm doing my job right. So our next steps then are to be, we're going to do a capture kit. So the DNA that you saw in the bioanalyzer result basically is all DNA in the sample. So we want to pull out only the human DNA. So this capture kit is going to basically target and pull out only human. For this, we're going to do mitochondrial DNA. Pull that out, we're going to sequence it, and then we'll data analyze and process. And from this data, we're going to be able to tell kinship, so how are these people related to each other? We'll be able to tell population ancestry, very similar to what Pat was talking about with contemporary population diversity in Africa. And then we're going to be able to compare this population diversity with that of contemporary African Americans and African descended persons in Charleston. So we're really excited to keep moving forward and keep checking through the project. And this is kind of what the suit looks like. And I just want to thank everybody for having us here. And I'll be happy to answer any questions you guys may have.
Please hold your questions, and are we ready to